examples of indicators. Okay, so what is indicator? Which is indicating the completion of reaction by a specific color change. Okay, so there are different kind of indicators. We have synthetic indicators, which is prepared in the lab by mixing certain chemicals. Olfactory means we have indicators which have a specific smell. So when it reacts with acid or base, it gives a specific kind of smell. We can identify with that. Universal indicator, which will be showing different colors in acidic and basic both. So some of the natural universal indicators are also there, like in our olden classes I have already mentioned, like purple cabbage. Purple cabbage gives huge range of color in particular medium when the juice of the or extract of the cabbage is treated. So when acidic substance it is treated, it will show the color with the acidic range pH, correct. Basic substance, when it is treated, it will show the color change like that, from pink, light pink, red to blue, dark blue and greenish, like that. So complete range of color change it is observing. It is treated with whatever substance, acidic or basic, it will be observing that particular color change. So that kind of indicators, which is same for acid and base, are called universal indicator. So let's see now few of the examples. We can see here there is one indicator called phenolphthalein. Okay, phenolphthalein. That indicator is always used in very few drops. When it is added to an acid, okay, this beaker has acidic solution, this beaker has basic solution. So in acidic solution, few drops of phenolphthalein, when it is added, we will see there is no color change, means. And when it is basic solution, phenolphthalein is added, then we can see the solution is turning pink. Means what is it indicating? It is indicating phenolphthalein is an indicator which shows color change with the basic medium. Okay, like we can see here also, pH 7 is a neutral pH. Water's pH is pH 7 and pH 10 above 7 pH is the basic pH. So here one basic solution is there, like sodium hydroxide, potassium hydroxide, like that. So in one conical flask we have water and another conical flask we have alkaline solution. When we add phenolphthalein indicator in both, we can observe it with water, it does not show any color change, and with basic medium it shows pink color. So phenolphthalein, what is our conclusion? Phenolphthalein gives pink color in a basic medium, like this. Phenolphthalein is a name of an indicator which you will be commonly using it in this school levels. There are another type of indicator called methyl orange. Methyl orange will show, see they have shown the color also. When methyl orange is treated with an acidic solution, when few drops of methyl orange is added, it will show pink color. Okay, but when it is added in alkaline solution, so it will show golden yellow color. So methyl orange is basically giving color change in acidic medium. Okay, and phenolphthalein is giving basically a color change in basic medium. So up to school level, up to 12th standard, you will be doing some of the experiment called titration. Titration means where the unknown solutions concentration we can find out, and by doing the titration using volumetric analysis using burette and pipette kind of apparatus. You will learn shortly about that also. So using that we can find out. So when because burette one apparatus, pipette is another apparatus, both the apparatus, the solution is filled is colorless. So to identify the completion of reaction, we are adding few drops of indicator in a conical flask where the experiment we are doing it, and when we start the experiment we will observe the color change. As the color change comes, we should stop the experiment and note down the reading. That will only indicate how much solution is required of other solution. Like one solution will be measured exactly, like 10 ml we are taking. So now from the burette how much solution is required to reach end point. End point means color change, end of the reaction, like this. So you are basically using up to 12 standard phenolphthalein and methyl orange indicator only. So phenolphthalein for basic medium, methyl orange for acidic medium. Now see, collect the following solutions from the science lab. Now you have to collect all these solutions and find out using treating with the different indicators. If you treat, you will identify the particular indicator gives color in which medium. Like hydrochloric acid is an acid, then sulfuric acid is also an acid, nitric acid is also an acid, acetic acid is a weak acid. First three acids are the strong acid. Acetic acid is a weak acid. Sodium hydroxide is a base. Calcium hydroxide is also a base. Potassium hydroxide is also strong base. Magnesium hydroxide and ammonium hydroxide are the weak bases. So few are bases here, few are acids, few are strong acids, few are strong bases, few are weak acids, few are weak bases. So how they show the color change? Let us observe that. Okay. Now here put a drop of each of the above solution on a watch glass one by one and test it with a drop of the indicator. Okay. Watch glass means it will be like a glass plate kind of thing. You will observe it in your labs. So there few drops will be added, and it will be added with the along with the indicator, 1 or 2 drops of indicator, and each one, like we have seen the list of acids and bases, so each one will be tested like that and observe the color change. That is our experiment. Now here also once again the same kind of thing we will see. Phenolphthalein shows pink color with the base. Okay. Methyl orange shows pink color with the acid. Then we have with the pH 7. pH 7 means with the neutral solution phenolphthalein does not show any color change. Okay. Here also methyl orange in alkaline solution does not show any color change. It is the it is showing golden yellow color only. Okay. Now these are the solutions which we have taken it. What kind of observations are expected from you when you do the experiment? Okay. So HCl, when red litmus solution is touched, no change. Blue litmus solution turns blue. It means what? Blue litmus turns blue means solution is acidic. Phenolphthalein colorless. Phenolphthalein shows color only in basic medium. This is an acid, correct? HCl. Then methyl orange, it turns red. It means substance is acidic. Second solution, sulfuric acid, that is also a strong acid. We have to observe red litmus solution, no change. Blue litmus turns blue, means substance is an acid. Colorless with phenolphthalein. Phenolphthalein gives color only with basic solution, and methyl orange gives solution with change in color with the acidic solution. So with acid, methyl orange turns red, means it proves it is an acid. Third acid, nitric acid, also no change with red litmus. Blue litmus turns blue. Phenolphthalein colorless again, and methyl orange turns red, means substance is an acid. Now acetic acid is a weak acid. So red litmus no change. Blue litmus again turns remains blue only. Then we have phenolphthalein again colorless, but here methyl orange turns orange. Okay, we can see the color change. Here it is red, here it is yellow, but here it is in between color, between red and yellow. Why? Because the solution is weakly acidic. Acetic acid is a weak acid. Okay, then sodium hydroxide is a strong base. So what happens now? Red litmus turns blue, means substance is basic by nature. Blue litmus no change. Then phenolphthalein shows pink color in basic medium. So sodium hydroxide is basic, so it is showing pink. Okay, and methyl orange does not show any color change because it is basic solution. So conclusion is basic solution. Now calcium hydroxide is another base. It is turning red litmus to blue, means solution is alkaline. Then blue litmus no change, phenolphthalein turns pink, and again we have methyl orange which is turning yellow only. Then potassium hydroxide is a strong base. Here again blue litmus, red litmus is turning blue, means strongly alkaline solution. No change in blue litmus color. Phenolphthalein is showing pink color, which is indicating a solution is basic, and methyl orange shows it is turning yellow. It is staying yellow only. Golden yellow means a substance is basic by nature, because methyl orange shows color change only in acidic medium. Magnesium hydroxide and ammonium hydroxide both are also, we can see, they are showing red litmus blue. They are not showing any change with blue litmus because both are basic. Then phenolphthalein, both are showing pink color because they are basic in nature, and with methyl orange it is same looking, staying as golden yellow only. Okay. Next we are saying there are olfactory indicators. Olfactory means, olfactory organs means the organs which are responsible for smell. Okay. So olfactory indicators, some substances which gives one type of color in acidic and a different odor in basic medium. Okay. They can be used to test whether the given substance is acidic or basic. Now such indicators are called olfactory indicator, which has a specific strong smell, and they give specific smell in specific medium and specific color in specific medium. Okay. We will see now what are the examples. Onion, all of you are familiar with the smell of onion. Vanilla essence, in all sweet dishes you are having vanilla essence, like bakery items you are having cake, pastries, as well as in many sweet items also you are observing. So that is having a distinct smell, correct? So all of them have characteristic odor. Okay, and we can identify that smell with the, all are quite familiar with them. These changes when some acid or base solution is brought in their contact, they can be identified. The result in these cases are not quite satisfactory, because every time 100% same results doesn't come which we are expecting it. Like we expect like phenolphthalein, methyl orange, which shows, phenolphthalein shows exactly pink color in basic medium and methyl orange shows pink color in acidic medium. So exactly we can identify acid and base separately, but here results are not very satisfactory, and sometimes it is deviation from the expected results, and that is why this cannot be taken as a 100% proof indicators for the experimental purpose. This is just a type of indicator where you can understand it. Yes, this is also showing a color change. Okay, so let's see the next one. These are the onion slices. Now they have small experiment. Onion odored cloth strips. What they did, some finely chopped onion slices in a plastic bag they have filled it. Okay, and some strip of, with some strips of clean cloth pieces also, some small strips are cut, that is also in the same plastic bag it is put. Then they have tied the bag tightly and put it into the refrigerator overnight. Okay, so the cloth strips can now be used. Now that cloth strips which are in contact whole night with that onion strips will be taken out, which has a smell of onion. Okay, will be taken out for the test of acid and bases. Then 2 of these cloth strips and check their odor. We can check their odor. Keep them on a clean surface, put a few drops of dilute HCl and on one strip, and another strip we can add a few drops of NaOH solution. Okay, so rinse both the cloth strips with water and again check their odor. After rinsing we can check their odor again. Note your observation. Okay, so onion smell is remains as it is. That smell doesn't go very easily. Now we can say with the acid or with the base, where the onion smell you are getting it. Okay, with the acidic substance, onion smell is observing more prominently. With basic substance, with this means when we add few drops of HCl and NaOH, that time we can see with the base onion smell is not very much prominent. So what we can observe it is showing a color, giving a specific odor with the acidic medium more. Now take some dilute vanilla essence and clove oil and check their odor. Clove oil also have a specific strong smell of clove. Take some dilute HCl solution to one test tube and NaOH solution to another. Few drops of vanilla essence to both the test tubes. We have added 2 test tubes are taken, in one HCl, in one NaOH. Both the test tubes we are adding few drops of vanilla essence. Shake well, check the color and odor once again and record the change in odor. Again we can record the odor, what kind of odor is observed. Similarly, test the change in odor of clove oil also with acid and with base and record observation. This is just at home level these substances are available, so you can also do this experiment and observe it. You will find out there is a different kind of specific kind of smell and with the acidic substance and smell with the basic substance. Okay. Then we have here, this is called pH range. Now this is a pH range, starts from 0 to 14. You can see how this pH strips when it is dipped into a particular solution of acidic or basic or neutral, it is showing this color change. So this is the strip of color, and we can match with this and we can identify the pH of the substance. Okay, so for 0 we have this kind of fire red color, for 1 acidic, very strongly acidic, orange, then weakly acidic, strongly acidic 2 also yellow, then greenish, it starts green. Now we can see here the weakly acidic will be greenish and 7 will be neutral, which is green color. Now as it goes, after 7 it will be basic. So as it goes this side, the green will start turning to blue, then blue will be blue 9 and 10, pH 9 and 10 will be little weakly basic solutions. As it goes to the stronger basic solutions, it turns to violet color. So 13, 14, 12 are very strong basic solution, so that is violet color. Okay, so more the pH, more basic solution, less the pH, more acidic solution, and 7 is a neutral pH. Mostly water has a neutral pH. Okay, now what is universal indicator? Universal indicator, we can see here when this one drop is touched with any kind of solution, acidic or basic, it is showing a specific color change. Okay, so universal indicator only tells us whether the given solution is acidic or basic, but we can also judge how strong acid or base. How we can judge? By the color change matching with the strip. If the color is purple, it means 14, pH close to 14, so very strong base. If the color is close to greenish, it means it is weakly acidic or weakly basic. Color is yellow, means very strong acid, something like that. Okay, then okay, so chemical definition of acids and base, physical properties of acids we are seeing now, how we can define acids and base. Substances on dissociation which gives H plus ions are acids. Substances on dissociation gives OH minus ions are bases. Okay, like for example, substances when dissolved in water give H plus ions, like HCl breaks to H plus and Cl minus. H plus ions are split into ions. When they split into ions, H plus ions are obtained, means it is an acid. Sulfuric acid H2SO4 gives 2H plus and SO4 2 minus, means it is also an acidic substance. Basic substances will give OH minus ion, like MgOH twice. When on ionization you get Mg2 plus and 2OH minus, so basic substance. Then KOH, K plus and OH minus, so this is also a basic substance. Substance when dissolved in water gives OH minus ion are called base. Okay, so valency of sulfate ion is 2, that's what SO4 2 minus. Okay, when dissolved in water, aqueous means when dissolved in water. Aqua word is related to water. So whenever you are making solution, see, solution can be made in different solvents. Water is also a universal solvent. Most of the substances dissolved in water. Okay, but some substances are not at all soluble in water. Those substances has to be dissolved in different solvents, like organic solvents are there, benzene, toluene, something like that. And apart from that, there are acetone, many other solvents are there. Okay, so solvents means they dissolve the substance into it, solute particles they dissolve into it to make a solution. Okay, they are called solvents. Universal solvent means most of the substances get dissolved, soluble in water, so that's what water is called universal solvent. But I am using the word most of, not all. Okay, because there are some substances which are not soluble in water. Getting it? So here when we write in a bracket, in a subscript, we write it AQ, which indicates aqueous solution. So which is very clear picture it is giving, we are preparing a solution in water only. Okay, now see here, classification of acids and base depends upon number of H plus and OH minus ions. Okay, like for example, the acid which gives more number of hydrogen ions in aqueous solution are called strong acids, like when they ionize completely to give lot of hydrogen ions, like HCl when ionized completely gives lot of H plus ions in the solution. H2SO4 gives lot of H plus ions in solution. HNO3 gives lot of H plus, complete ionization. Complete molecule will convert into ions. So they are called strong acids. Weak acids which are partially ionizing. Partially ionizing means gives less number of hydrogen ions in aqueous solution are called weak acids. Like acetic acid, when it breaks it gives H plus ion, but part of the acetic acid only dissociates. Part is remaining undissociated in the solution. That is called partial ionization. Okay, so because it ionizes partially, so number of ions are produced by that is less in the solution, so they are weak acids or weak base. Okay, so weak acids, weak bases are partially ionizing. Strong acids, strong bases are completely ionizing. Right now we are talking strong acids and weak acids. Now all mineral acids are strong except carbonic acid. Carbonic acid is weak acid. When carbon dioxide gas dissolved in water, you get H2CO3. H2CO3 is carbonic acid. Okay, I can write formula here if you want, just for your reference. All organic acids are weak acids. Okay, a dilute solution of weak acid are quite safe to drink, because when water quantity is more, so acid will not cause any harm inside the stomach or intestine. Okay, hence they are used as food ingredients. Most of the food ingredients like lemon juice, tamarind, which are all are acidic in nature. Here we can see how the dissociation of strong acid. I have told you strong acid which ionizes completely. Now ionization complete means, for example, HCl. So there are plenty of HCl, H plus and Cl minus ion in the solution. So it means the solution is of the strong acid. Okay, now one more rule you should follow for the strong acids: always add acid to water. Whenever you are diluting the acid, always acid should be added to water. Why? Because otherwise if you add water to acid, acid will spurt out, because it is an exothermic reaction, lot of energy generated, and that acid can cause a disastrous reaction. When it spurts out, it can hurt, it is highly corrosive, so it can hurt your body or your clothes. It get some kind of small holes or some kind of reactions with that. So that is why you have to be very careful. You have to be, whenever you are making a solution, add acid to water. Remember this. Okay, this is very important. So clear it is. What is strong acid? Strong acid completely dissociates in water. So because of complete dissociation, there will be plenty of ions in solution. In aqueous solution. Okay, aqueous means solution in water. Now they undergo incomplete ionization in aqueous solution. So because of that, what happens? The number of ions will be less. Why? Because their reaction is incomplete. Now we can see here there are, first we will complete this. So if we say acetic acid breaks to CH3COO minus and H plus. So number of ions, because undissociated molecule is also present in the solution, that is why complete dissociation is not taking place. So the number of ions are less. Okay, so due to less number of ions present, incomplete ionization takes place, and that is only called a substance is weak acid. So this is a tendency of strong acid. Okay, and this is a tendency of weak acid. Clear it is? So next we will see few properties of acid. What are the properties? Now we can understand chemically by experiment purpose. Also we have done lot of experiments we have done. So acids have sour taste. That is like we have seen the daily life examples like lemon, tomato, tamarind, all these are sour to taste. Then acid turns blue litmus to red. Whenever substance turns blue litmus to red, that is called acid. Red litmus to blue, that is called base. Acid solution conduct electricity. They are electrolytes. Like we can have acidulated water. Water will not conduct electricity in presence of, means the distilled water will not conduct. If there are dissolved salts are there, like a tap water or few drops of acid when we add, and then we pass the electric current, it is providing passage for electric current because it undergoes ionization, and lot of ions are providing passage for electric current. So they are very good electrolytes. Electrolytes means the liquid which is in form of ions, which is conducting electricity. Then acids reacts with metal to form hydrogen. Always any acid, when it reacts with any kind of metal, what will happen? It will separate hydrogen gas, it liberate it. Like for example, we can see metals plus acid gives metal salt plus hydrogen gas, like zinc metal, magnesium metal, sodium metal, all will show this kind of reaction, and you are getting a salt and hydrogen gas is liberated. Okay, so different type of metals we can say, zinc, Mg, sodium will liberate hydrogen gas with acid. When they reacts with acid, what they give? They give hydrogen gas. Okay. So sodium, you can see the piece of sodium when it is added in acid in the test tube, it will start moving very fast, that piece. Why? Because it is an exothermic reaction, as well as hydrogen gas will start bubbling, and that will be heat is generated, so that piece will start moving very fast and the gas will start bubbling up. Okay. Now we can see this is the litmus test also for acid. What is the litmus test? Acids will show a litmus turn, acid turns blue litmus to red. So they are adding blue litmus. When it is touching to the acid part, it is turning red. It is indicating the substance is acid present in this beaker. Okay.